thank you very much for inviting me here. So I'm going to talk about aposematism and mimicry in butterflies and causes, consequences, and paradoxes. So insects may well become our main protein source in the near future if they don't disappear. But in fact, insects already have many non-human predators, and as such, a range of anti-predator defenses have evolved, such as crypsis, wow, it's difficult, masquerade, wherein you look like a non-edible item, or even a scary one, deflection of predator attack towards non-vital parts of the body, or intermittent flashes of colors that confuse predators. But these are all deceptive adaptations, and another way to deter predators from attacking you is to have chemical defenses and be unpalatable or toxic. And many chemically defended prey have also evolved conspicuous color patterns that advertise unpalatability to predators, and this is called aposematism. So the way it works is that a naive predator attacks and eats a prey, and then it finds it's really disgusting, like this poor jay here. And then after a few trials, the predator learns to associate the color pattern with the unpleasant experience. So it's based on a learning process. And because the probability of encountering a naive predator decreases with the abundance of a given color pattern, there is strong selection for convergence on a same color pattern across co-occurring prey species in communities. So for instance here, these birds have already encountered species 1 and knows that it's not good. So it will not attack species 2 and species 3 because they look like species 1, but it will attack species 4 because it doesn't look like species 1. And so this was first described by a German naturalist, Fritz Müller, in the 19th century. And so this is therefore called Müllerian mimicry. And here, so these species are mimetic species, and individuals of different mimetic species interact positively because they benefit from the presence of one another. So there's a strength in number. And so species that harbor the same color pattern are called co-mimetic species, and they form what we call mimicry rings. And so please notice that, again, all species are toxic here. So there's another type of mimicry called Batesian mimicry, where some palatable species mimic unpalatable aposematic species. But I'm not going to talk about that here. I'm going only to focus on Müllerian mimicry, where all species are toxic. And so Müllerian mimicry exists in a wide range of organisms, but it's particularly common in insects and best known in butterflies. And two groups of butterflies have attracted considerable interest in mimicry studies. It's the genus Heliconius and the tribe Ithomini, which is probably the largest mimetic radiation. And so both clades are endemic to tropical America, and they're both chemically defended with cyanogenic glucosides for Heliconius and pyrrolizidine alkaloids for Ithomini. And they're both aposematic and engage in mimetic interactions with other species. So as an example, here are two Ithomini species that have diverged 14 million years ago. And here are mimicry between Heliconius and Ithomini species with 82 million years of divergence, so the same time as divergence between human and mice. So in this talk, I'm going to go through some major advances on mimicry studies. First, I'm going to talk about the puzzle of mimicry diversity, then the link between mimicry and speciation, the formation of species, then the genes that determine mimicry, and then the ecological implication of mimicry. And finally, I'll finish with another mystery, the evolution of transparency in mimetic butterflies. So first, the puzzle of mimicry diversity. So as I said, there is a strong selection for convergence on the same color pattern across many co-occurring aposematic species, but yet there is an awkward diversity of mimicry patterns in mimetic butterflies. So if selection is so strong, why don't they all converge onto the same color pattern? And in fact, there is diversity at several levels. So first, there is diversity among regions at a wide geographical level. So for instance, here you have two different species, one on the top, another at the bottom. And you see that they're very similar to each other in each region, but they vary in a concerted way in different regions. So that's one thing. But you can even get diversity within a locality. So here you can have different species that belong to different mimicry rings in the same place. And even worse, you can have within species and within locality diversity. So first, what about diversity among regions? So here is a geographical representation of the variation of color patterns in these two species. And again, you can see that they co-vary in a concerted way, and they form a spatial mosaic of wing color pattern. And an explanation for that is that the communities of predators are actually different in different regions, such that there is very little selection for convergence on color pattern across different regions. And this hypothesis was confirmed using a modeling approach, where the distribution of two aposematic species was simulated with the segregation of predators. And so you can see that the output is a nice mosaic that is very similar to that one, and that the two species have a similar mosaic of color patterns, so mimicry has evolved here. I just forgot to say one thing. So I said that there were two main clades of butterflies that have been studied in mimicry studies, Heliconius and Ithomeni. And so because they have different particularities, Heliconius have mostly been studied for developmental biology and genetics, while Ithomeni have been studied mostly for ecology and diversification studies. And my own work has mostly focused on Ithomeni, but in this talk I'm going to extensively present also results on Heliconius from my colleagues. Right. So okay, at the regional level, we understand why diversity of color patterns can be maintained. This is due to predator turnover with geographic regions. Okay, now let's go to a more trickier situation, local diversity within species. So here we have this Heliconius species, Heliconius Numata, which is polymorphic within population, meaning that if you go to a single place, you will encounter individuals of the same species in the same population, but that have different color patterns. And in fact, Heliconius Numata engages in mimicry with several Ithomeni species here, and the distribution of Ithomeni species and color patterns is a mosaic, as we've seen before. So that's very classical. But now, if you look at the distribution of Heliconius numatamorphs, then you can see that although they tend to co-occur in the same localities as their co-mimetic species, the distribution is wider and largely overlap, such that there is diversity in different localities. And so this is due to the wider mobility of Heliconius numata. And so this produces the local polymorphism. But this species is very particular, and it's really not the rule to have that. But what is really pervasive is to have within community diversity of mimicry rings in different species. And so, for instance, in the hyper-diverse forest habitats of Peru or Ecuador, you can find up to 15 different mimicry rings co-occurring like in a very, very small area. So an explanation to this has been hypothesized, and it's that suppose that a piece of forest is actually a mosaic of microhabitat, for instance, understory and canopy, and if predators are segregated by microhabitats, then again, there is very little selection for convergence on color pattern across microhabitats. And so these predictions were confirmed with a theoretical model. So we wanted to test these predictions in the field by surveying both butterfly and their potential predators, insectivorous birds in that case. And so we went to a place in Amazonian Ecuador, Anyango River, and so we surveyed a very small area, 3 kilometers square, and we also conducted predation experiments. So this is how the forest looks like there. So you can see that there is a diversity of microhabitats, so of course, understory and canopy somewhere there, but also little streams and ridges, which are very, very close apart, open and closed forests, etc. And that's the way we sample the butterflies. So we use nets, and sometimes they have to be very long. We also climb trees to access the canopy, and then we mark and release butterflies after identification, because it's very difficult to identify them in flight with mimicry. And birds have been surveyed with binoculars. So there we found 58 Ithomeni species distributed among 8 mimicry rings and 176 insectivorous bird species. We also recorded topography, phrase structure, flight or resting height for each butterfly and bird individual, and we conducted predation experiments in different microhabitats. And so our results show that there is microhabitat segregation of predators and mimicry ring, and also that in our predation experiments, we swapped different species in different microhabitats, and species that are put in the wrong microhabitat are much more attacked than in their own microhabitat. So this segregation of predators and mimicry rings explains the stable mimicry diversity in those communities. So to conclude about this apparent paradox of diversity, mimicry diversity, in fact, now we understand better why it's possible to maintain such a diversity at different ecological and geographical levels. So now I'll move to the next point, mimicry as a driver of speciation. So speciation is the evolution of reproductive isolation among groups of species, and mimicry has been put forward as a major driver of speciation in mimetic clades. And indeed, so experimental studies have shown that there is strong assortative mating for a wing color pattern. So you remember that in Heliconius and also in Ithomeni, you may have different geographical subspecies with different color patterns. And so this guy here always prefer a mate that looks like it rather than another subspecies. And this is purely based on color pattern. And even if, so this induces pre-mating reproductive isolation. And even if those guys manage to mate, then they produce a hybrid that has an intermediate color pattern which is not mimetic and not recognized by predators as toxic. So this will be heavily attacked, and this again has been shown by experiments. So this induces post-mating reproductive isolation. So yeah, shifts in mimicry pattern causes reproductive isolation and speciation, and this is mimicry, is thought to be the main driver of the diversification of Heliconius butterflies and one of the main drivers of the diversification of Ithomeni. So I hope I've convinced you that mimicry and aposematism are fantastic adaptations. And so what are the genes that underlie color pattern variation? So all the advances come from the Heliconius studies. And so in Heliconius, a handful of loci called the Mimicry toolkit have been found. So these are homologous loci, and they've been found to control color pattern variation in many different Heliconius species. So note here that this is the Heliconius Numata, the polymorphic species, and in this case, all variation is controlled at a single chromosomal region on a single chromosome. And so what are these genes? So some of them have been identified and revealed with expression pattern analysis during the wing growth during the pupal stage, and also more recently CRISPR-Cas9 KO. And so you can see that these genes are not genes that they're not involved in the production of pigment, but instead those genes are determining the fate of the cells, and so they act as switches between different color patterns. And so this mimicry toolkit in Heliconius can produce patterns that are similar or different. And so for a long time, it has been thought that convergent color patterns in Heliconius were produced by parallel evolution, meaning that the same genes were recruited independently in different species. But more recently, it has been shown that in some cases, some color pattern loci, like those red loci here, have introgressed from one species to other species. And so this has been shown in several cases, and in particular, in the case of the polymorphic species Heliconius Numata, where there has been integration, but only in some of the morphs, not all of them. So I'm going to speak a bit more about this species because it's really weird and very unusual. So as you remember, Heliconius Numata harbors different morphs, and each of them mimics a distinct species of Ithomeni butterflies. And so in other Heliconius species, so we have different geographic subspecies with different color patterns, and as we've seen, they produce hybrids with intermediate color patterns, which is not recognized as unpalatable, so which is attacked by predators. But luckily for them, this only occurs in very narrow hybrid zones. But the situation is much trickier for Heliconius Numata because in all population there are multiple morphs that coexist. So how come that individuals with intermediate color patterns are never found? And part of the explanation might lay in the special genetic architecture of the control of mimetic pattern, which is concentrated in a single region called a supergene because it contains multiple genes, such that it limits. I mean, there is no possibility of mixing of alleles during the meiosis between chromosomes. But this region is quite big, 450 kilobases, and it contains many genes, so there is still possibility of recombination within the supergene. However, this hardly happens because, in fact, so different morphs are characterized by different haplotypes, and those haplotypes have different inversions of pieces of chromosomes within the supergene. And this impairs recombination. Moreover, there is a strict series of dominance between color patterns such that hybrids between individuals that have different color patterns always look exactly like one of the parents. They're always mimetic. So that's great, but if all your adaptive variation is controlled by a single region where a recombination is impaired, then there's a serious risk of accumulating deleterious mutation. But one way to get around that is to try to maintain this deleterious mutation at a heterozygous state. And so this should select for individuals to mate with other individuals that have a different haplotypes than their own, meaning that they should try to mate with individuals with a different color pattern. So this should select for a disassortative mating, unlike other Heliconius species which show assortative mating. And so this sounds crazy, but actually this is what my colleagues found. So for many Heliconius morphs, there is strong rejection by females of males that harbor the same color pattern, while they readily accept males that are different. So to summarize the fabulous tale of Heliconius Numata, so there is local polymorphism in population, and this selects for a supergene architecture with a strict series of dominance and little recombination due to inversions. But this, in turn, selects for disassortative mating, which maintains polymorphism, and so on. So now I will move to more ecological studies. So how mimicry can affect the evolution of the ecological niche. So I'll go back to my diverse community of ethomini. And so you remember, we've seen that there is microhabitat segregation of both predators and mimicry rings. And so the fact that mimicry rings are segregated by microhabitat could stem for two evolutionary scenarios. Either that all these species here have inherited both their color pattern and microhabitat from their common ancestor, or that there has been adaptive convergence for both color pattern and microhabitat, so selection. And so by conducting comparative analysis using the phylogeny of this species, we showed that actually the adaptive convergence scenario is much more likely. And this was also predicted by a theoretical model. But we can go even further, because butterflies are phytophagous, and actually they're pretty specialist on their host plant. And if host plants are also segregated by microhabitat, then we would expect co-mimetic species to also share larval host plant. But if segregation is not so strong, then instead they would benefit from diverging on host plants such that competition is minimized. And so again, using a theoretical model, we found evidence for both convergence or divergence on host plant depending on host plant distribution. And this was also confirmed in a field study. So the ecological niche of mimetic butterflies is actually strongly determined by mimetic interactions. And so now I'm going to my last point. I think I've spoken very quickly. The mystery of the evolution of transparency in mimetic butterflies. And, well, so you remember aposematism means advertising unpalatability using conspicuous color patterns, and it has been shown in experiments that increased conspicuousness increases the learning rate and the memorability by predators. So it's beneficial to have conspicuous color patterns, and indeed, many aposematic and mimetic species do have conspicuous and contrasted color patterns. Yet, ethomini butterflies, the largest radiation of mimetic butterflies, have many species which have transparent color patterns. And transparency is usually associated with reduced detectability, for instance, in marine organisms. So if this is the case here, why has transparency evolved in an aposematic group of butterflies? Well, but the first thing, we need to confirm whether transparent species in ethomini are indeed less detectable than opaque species by predators, because although they're transparent, they also have opaque elements. And if we find this is the case, then perhaps this is because they're kind of giving up the aposematic strategy to switch to crypsis, and perhaps this is because they're less toxic than opaque species. So to investigate the first question, then we conducted detectability experiments, so using real predators that had to find 4 species that spanned a range of transparency. And we complemented these observations with real predators with modeling detectability by predators of a larger number of species. And so both experiments gave the same results, meaning that we found that transparent species are less detectable than opaque ones. So now we can move to the second question. So if they're less detectable, is it because they're less toxic? And so to answer this question, we conducted taste rejection experiments using chicks. And so they were fed with pellets and with different colors, and one color had butterfly in it. And so this enabled us to assess the unpalatability of different species. We tested 10 different species, and then to correlate unpalatability level to transparency level. And here we find that all transparent species that were tested were highly toxic and unpalatable. They were strongly rejected by chicks, whereas we find a range of unpalatability in opaque species. So actually, this strongly rejects our hypothesis. And not only transparent species are no less toxic than opaque ones, but they might in fact be even more toxic. So, well, then we need to find an explanation for that. And the explanation we've come up with is that perhaps those species are making the best of both worlds. So this is a French private joke. And meaning that because they're transparent, then they're less detected, and so they suffer less predation, especially by predators that are not sensitive to their toxins. But if they're detected, then they're identified by experienced predators are unpalatable, and they're not attacked either. And the fact that all these species, the transparent species, are highly toxic, probably stems from the fact that there is a compensation between conspicuousness and unpalatability. So conspicuousness increases the learning rates and memorability, but unpalatability also does the same thing. So if you reduce conspicuousness, then by maintaining high unpalatability, you can still have a strong learning rate and memorability. So to conclude, mimicry is a compelling example of the power of selection, because not only it has mimicry implies the evolution of chemical defenses, conspicuous color patterns, and convergence of these color patterns among co-occurring species, but also consequences can go as far as modifying the genetic architecture and dominance, and also they can also have evolutionary implications on the niche of the species, including host plants. And we've seen that mimicry is probably a strong driver of speciation, and because mimicry is linked to habitat and to host plant, then shifts in mimicry might incur shifts in habitats and host plant that might in turn drive increasing reproductive isolation. So it actually may have cascading effects on speciation. And then, so mimetic species engage in positive interactions between each other, and so we can see communities are tightly co-evolved assemblages, and this raises the question of the responses of such communities in the face of global changes, where a community might be disassembled and positive interaction lost. And finally, so now we have a pretty good understanding on why there is stable maintenance of diversity in mimetic systems, but we still don't know where the origin of diversity comes from, because this could all work very well with a single color pattern. And so why do novel color patterns seem to appear so readily, and how can novel color patterns become established? So recent theoretical advances have proposed some mechanism that could help rare color patterns to be maintained, such as neophobic behavior by predators, but they need to be tested with empirical experiments. And finally, so there's the puzzle of the evolution of warning signal conspicuousness in aposematic butterflies. So we've determined that it may pay to be both aposematic and unconspicuous. And so the question is perhaps why has transparency evolved in some aposematic butterflies, but rather why has transparency not evolved in all aposematic butterflies? So I'd like to thank all people that contributed to this work, including my colleagues working on Heliconius, funding sports, and people in the field. And thank you very much for your attention and for the Académie of Sciences to host me today.
Thank you. So we have time. We have time for 1-2 questions before the photo. Who? Joanne.
I was wondering if there is good evidence for positive selection for the mimicry genes. Do you see the genes that determine the color patterns? Are they under strong positive selection?
So I don't know very much the evidence for this. I think there are studies, yeah, that show that they're under positive selection. But so most of the studies were more like functional studies, developmental studies. But no, yeah, I think that, yeah, there are some papers that show evidence for positive selection. Yeah.
Other questions? Elena.
Wonderful talk. I would like to know how toxicity is linked to the color code. So is it the toxic compounds are in their wings and in the color, or what makes these butterflies toxic?
I'm not sure I got everything. So you ask whether how the toxicity is linked to color pattern, or in which sense? In like a physiological correlation, or because apparently there is, well, as far as we know, there is no physiological correlation between toxicity and color pattern. So these are like distinct molecules, and but as we've seen, there seems to be an evolutionary correlation. Of course, this needs to be confirmed with more species, but apparently the less conspicuous species seems to be more unpalatable. There's a huge body of literature on honest signals and with different predictions, so a lot of models, but some models predict that signaling should be honest and others don't, but it depends also on the assumptions of those models.